Assalamu alaikum students. I hope you all are fine and safe at home. I'm fine too. This is week 9 and lecture number 1. We have started the chapter number 6, that is about the enzymes, and the topics are the factors affecting the rate of enzymes and the mechanism of enzyme. Here are some objectives to summarize the factors that affect on the rate of enzyme and describe the mechanism of enzymes. First of all, we will discuss the factors affecting the rate of the enzyme action. In last lecture, you have already studied about the characteristics of the enzymes. Either they are very specific in nature, they affect on the rate of the reaction, and the temperature is also affect on them, pH is also affect on them. So we can say that the enzymes are very specific in nature and they are very sensitive to the environment in which they work. Any factor that can change the chemistry or shape of the enzyme molecule can affect its activity. Some of the factors that affect the rate of enzyme action are discussed here. First of all, temperature. Increase in temperature speed up the rate of enzyme catalyzed reaction, but only to some extent. Every enzyme works at its maximum rate at a specific temperature called optimum temperature for that enzyme. So, the optimum temperature is that temperature at which the enzyme acts. When temperature rises to the certain limits, heat adds in the activation energy and also provides the kinetic energy for the reaction. So the reactions are accelerated. But when the temperature is raised well above the optimum temperature, the heat energy increases the vibrations of the atoms of enzymes and the globular structure of enzyme is lost. So this is known as denaturation of enzyme. It results in the rapid decrease in the rate of enzyme action and it may be blocked completely. So, students, if the temperature rises to a certain level, it means that the optimum temperature level, it may increase. So, the enzyme action is increased and the reaction rate is also accelerated. But if above the optimum temperature, if heat is given to the reaction, what happened? The enzyme activity is lost because the enzyme is denatured in its activity. Its structure is lost, so we can say this is the denaturation of the enzyme. Or sometimes the enzyme action is decreased and also may be blocked. So a very specific amount of temperature is required for every reaction. Here you can see temperature and the rate of enzyme catalyzed reaction. At optimum temperature, enzyme increases the rate of the reaction, but after that, if we will increase the temperature above the 40, 50 or 60, the enzyme become denatured or its activity is lost. You can see it's very clear from this diagram. Now the substrate concentration. If the enzyme molecules are available in reaction, increases in substrate concentration increases the rate of the reaction. If enzyme concentration is kept constant and the amount of substrate it increase, a point is reached where any further increase in the substrate does not increase the rate of reaction. When the active site of all enzymes are occupied at high substrate concentration, any more substrate molecule do not find free active site. So the students, where the active sites of the enzyme bind to the substrate and there is no active site free for any substrate, then in this condition, if we'll increase the substrate concentration, there will be no effect on the rate of the reaction. So at specific level, the reaction rate increase but a specific level. Here in this diagram you can see if we will increase the substrate concentration at specific level, all the sites of the active sites of the enzyme become occupied, so more increase in the rate is not possible. It become constant. Now the pH. All enzymes work at their maximum rate at a narrow range of pH called the optimum pH. A slight change in this pH causes a retardation in enzyme activity or block it completely. Every enzyme has its specific optimum pH value. For example, pepsin working in the stomach is active in acidic medium, low pH, while trypsin working in small intestine shows its activity in alkaline medium. So it's very specific for the enzyme they work at, which either at acidic pH or alkaline pH. So it is specific for every enzyme. If for enzyme that works in alkaline medium decreases the pH to acidic, then it will not work or its activity will block. Here you can see at optimum pH it will work. After that, if we will change in the pH, then what happened? Enzyme become denatured or its activity will block. Okay, mechanism of enzyme action. When the enzyme attaches with the substrate, a temporary enzyme substrate complex is formed. Enzyme catalyzes the reaction and substrate is transformed into product. After that, the enzyme substrate ES complex breaks and the enzyme and the products are released. Here is the lock and key model. According to this model, both the enzyme and substrate possesses the specific shapes that fit exactly into one another. You can see in this diagram, here is a blue in color enzyme and the substrate. Both fit into each other like a lock and key. Like key fits into the lock, like this, substrate fit into the enzyme. The active site of the enzyme occupied by the substrate. So in this way, what happened? The product forms as the result. In this diagram, you also can see the ES complex or enzyme substrate complex. Here in this diagram, the enzymes have 3 active sites and there are 3 substrates, so they can bind to the enzyme and the product is formed. Okay, here is the assignment. You have to write a note on the mechanism of the enzymes and you have to prepare your chapter number 6 for the upcoming test. In the next week lecture, we will discuss the whole chapter review and marking the short and long questions. Okay, till then, Allah Hafiz. Thank you very much.